Welcome to Microscopy 101, the first of a series of STEM events that are brought to you by Jump Simulation, University of Illinois College of Medicine, and OSF Healthcare. This is the first, like I said, of a series of microscope labs where we'll teach you how to use the microscope, we'll help you unpack the microscope, we'll help you learn how to build and create and explore on your own using a microscope, something that a lot of people don't think about as an everyday item, but really, if you're into any kind of science, the microscope is as useful as a hammer or a screwdriver, because it doesn't matter what field of science you go into, whether it's biology or chemistry or physics, any specialties like medicine, you're going to use a microscope at some time. I remember the time that I spent in high school and undergraduate and even grad school, all the microscopes I used throughout the years, when I didn't think I would ever be using them again, and I do. In fact, there's a table full of microscopes right behind me that I use. Some huge ones, giant ones, little ones. And what's really cool is that the microscope that you have today is one that I picked out that I found was perfect for the at-home explorer. Somebody that doesn't have to have a super strong microscope, but wants to see a lot of cool things in detail. And that's what we're going to do. Over the next 3 videos or 3 sections, we're going to bring you Microscopy 101, which is unpacking, getting to know some of the tools and the basic skills to think about while you're doing microscopy. The second one, 102, we're actually going to start building some slides, working with a lot more slides. And 103, we're going to take it a little bit more advanced and help you to come up with your own experiments and get going with all the tools. So let's us get going. Okay, so the first thing you want to do is you want to open up your microscope and you got a box just like this, the microscope box inside of it, and a lot of other items. So you'll take your microscope out, sit to the side for now. I've actually already unpacked mine, and I found this really inexpensive $6 toolbox. It's plastic. It's perfect for my microscope. So I'll be pulling everything out of here. You can see on the top of it all of the pieces that came with the kit. Inside, some of my fun tools and things I experiment with. Then the book and microscope. So let's start with the book. We're just going to flip through page by page, work with some of the items, and explore. So I put mine in a binder. Yours will come just like these, in individual inserts. I just cut mine out because I like the way that I can rearrange pages and flip through it, or if I decide to change the order or I want to take a recipe out of here and put it in another section, I totally can. So, the kit. Not sticky enough pages, fingers. So the first thing, and I always start off my STEM events this way, is that I want you to think like a scientist. I want you to think like a baby, actually, not a scientist, because scientist has a stigma. It's like, you know, person wearing glasses and a lab coat. But really, babies are probably the most brilliant scientists out there because they're the most inquisitive of all of us. They want to know about the world. They want to know what's going on. They don't understand it. It's a strange place. Just imagine if you were picked up and put on another planet full of all these, you know, creatures walking around and things crawling on the earth and all the cool, bright colors. You'd really want to absorb everything, and that's what babies are. When they come out, they don't know how to use their tools and equipment. They don't know how to use their eyes properly or ears or mouth or anything. So they want to touch and experience everything. I really want you to be that way anytime you do any kind of STEM event, is experiment. Maybe not taste everything or stick something straight up to your nose, but be like a baby. Wonder why it's the way it is. Wonder what's missing. So what's cool about microscopes is that you're seeing a world that you've probably never seen for the first time. When you look at anything simple like a piece of paper or a dollar bill, things you've maybe handled a million times in the past, but you've never really thought about it. So we're gonna look deeper into this new planet or this new world, and I really want you to take it the same way a baby would take it, exploring and understanding. Look at things, look at the details, look at how things are shaped, look at the different colors that maybe you didn't see before. Just really get into this new world. I put a lot of QR codes on here as little extra things you can do to explore, but this QR code is a link to a set of studies about babies as scientists. So it's really cool to watch and see how they think and how they're naturally a scientist when we don't, you know, think about being a scientist. The next thing is you really want to understand the steps of the scientific method. And the scientific method is really hundreds of years old. It goes all the way back to the earliest investigators that we know. We've looked at people observing things that happen in the world. We've looked at how they question things. We've looked at how they build an experiment that's a safe, strong experiment that can prove what they think, and then how they share their data. And really, the scientific method is something we do every day, whether you realize it or not. You observe things. You might notice something about the foods that you eat, like a certain food is better when it's warmed up, or it's better when it's cold, and you think, I'll try this in the microwave, or I'll try this after I put it in the freezer, like yogurt. I mean, recently we've been experimenting with yogurt sticks for kids. We've been putting them in the freezer and never thought of that before, but they're delicious and kids love them. Who figured? I tried it in the microwave. Yogurt in the microwave is disgusting. So you observe, you question, you wonder, you create some kind of an experiment. What if I do this? So when you're doing that, you're creating a hypothesis. It's what if I do this? Will I expect this to happen? So I've observed that yogurt's pretty good when it's chilled in the refrigerator. My question is that if I make it colder, will it be better? My hypothesis is yes. If it's colder, it'll have that creamy, that creaminess of yogurt, but it'll also have that frozen goodness like ice cream, but maybe a little bit healthier. So my hypothesis is yes, I'm going to really enjoy it. The experiment is put it in the freezer, pull it out, try it, compare. What do you think? When you're trying it, comparing, and telling yourself, what do I think? That's actually your collection, your data. And then what do? Modify and repeat. I thought, well, if it's cold and it's really good, what if I warm it up? Not so good. So I repeated, modified, and I found a totally different result. So as we're going through this, I really want you to think about how can you create things quickly. It doesn't have to be, a week-long experiment or anything like that. It can be just as simple as, I've noticed this thing. I wonder if, I bet if I do this, and this is what I'm going to do. And then you find out results. Right? As a scientist, it's really important, or anybody interested in science in general, it's really important to have the right tools. And the very first tool is actually right between your ears. It's your brain. And just like this little guy here, the more you flex your brain, the stronger it gets. So as somebody that's asking questions or wondering what my world's like, it's really important that you ask a lot of questions, not just once or here or there, but the more questions you ask, the more you flex in your brain, the more questions you'll generate. Edison actually had the Edison quota, where he would have to come up with a certain number of questions or ideas in a week, or he wasn't satisfied. So maybe it was 100 new ideas in a week, or 100 new questions. And what's interesting about Edison is he also holds the record for the most patents ever. And it's probably because he had that inquisitive mind. It may be hard. You think, oh, questions are so hard. But what you'll find is when you flex your mind, it's like a muscle. If you ask yourself a question now and you ask yourself another question and another question, the more questions you ask, the more questions you'll come up with. Your brain's really good at answering questions and coming up with ideas. If you ask yourself a question like, oh, why is I hate to say this, but oh, when you're frustrated after testing, why am I so dumb? Your brain will actually answer that question. It'll come up with reasons. It'll go, well, you're not studying hard enough, you're not doing this. And it's usually things you don't want to hear and you get upset with yourself, right? Your brain works the same way with things you want to solve. If you really want to figure out how to build some kind of contraption so that your sister can't get into your room, your brain will come up with all these ideas. Will take those ideas and build something really cool and creative. The more times you do that, the better you get at it, the more creative questions you come up with, and the cooler the ideas you get. So the first tool you have is actually in between your ears, and it's your brain. You really need to use it. Be inquisitive, ask questions. The second thing is your lab. And before we actually open up your lab set, we want to go through some microscope safety. These are general safety rules when you're working with your microscope or any of the tools that are in the kit. So before you touch anything, let's walk through them. So first, always work in shouting distance of an adult, whether it's an older brother or sister that's responsible, or your parents or grandparents, whoever. So that if you do hurt yourself, if you cut yourself on a slide or you do something, you can shout for them and say, hey, I cut myself. Me, if I cut myself and start to bleed, I need to just sit down and relax. And I usually tell the person nearest to me, can you get me a Band-Aid? Because unfortunately, I have problems with seeing my own blood. There's my big weakness. So always be within shouting distance of somebody that can help you. All right. Maybe it's a good idea to keep Band-Aids. In fact, I would always keep a little running list of things you need, like a pencil, which is right here at the top of my toolbox. So things that don't come in the kit that maybe you want to gather, first thing would be Band-Aids. So if you do cut yourself, you can clean it up and move on really quick. The second thing, keep a trash can, sharps cup, or paper towels, paper towels near you, because you never know when you're going to spill. If you're working with pond water or whatever, you don't want to use expensive cleaning tissues to do that. You just want a paper towel nearby, something you can blot things up with or clean it up. So the next things you want to write down is you really want to have something to keep sharps in. Simple cup is handy. So if you break a piece of glass, you can drop it in there. You want to have a garbage can so that you can just push all of your debris. You're going to see me pushing things into the garbage can all throughout this. And you want to have something like towels, like disposable towels that you can throw away, because you'll work with stains. And if you get the stains on the towels, your family probably will be upset with you. So Band-Aids, a sharp cup for sharps, a garbage can, and some towels. Next, never eat or drink around your equipment. If you're working with pond water, you never know what's in there. It could be some kind of bacteria or something that can make you sick. So you don't want to be drinking your soda or eating a sandwich where you might have these invisible germs on your hand. Make sure you're not eating around stuff. Handle chemicals and specimens with care, using potential caution when you need to. If you're working with something that could make you sick, number one is question why you're working with that. Number two is that make sure you're wearing gloves, an apron, goggles. There's nothing that we're going to be working with that can make you sick. So we didn't put those in the kit, but if you are working with some nasty, gross pond water and you're curious about what's in this that's making it so stinky, wear some gloves, wear something to protect the splash. If you're working with a lot of stains for a long time, make sure you wear some old dirty clothes or at least an apron. All right, next, keep 2 hands on the arm and the base at all times. And this is talking about the microscope. So I'm going to open mine up. I'm just going to show you as an example. Actually, I have one right here. So here's the arm where my hand is here. Here's the base on the bottom. Always carry it with 2 hands so you don't drop it and risk damaging it. And then we'll pull yours out in just a few minutes. Keep your microscope covered at all times. Well, all times when you're not using it. So when you're not using it, it comes with a protective cover, but you don't want dust to get into the eyepieces. You don't want dust getting down into the objective lenses. Dust is actually more dangerous than you think to a microscope, because even small particles of dust, and we'll see looking at the microscope how significant this is, but if you wipe it or you brush the dust over it, it can be hard. It can be like microscopic crystals that are scratching your lens. So make sure you're keeping your microscope covered whenever you're not using it. Always start the stage. A little preview. This thing's a stage. Always start at the furthest away from the lenses. Okay. So that you have a working space, and then before you actually look in, I'll tell you about this, you're gonna bring it as close as you can to the lenses. Right? Never take your microscope apart. There are a couple times I'll ask you to take a lens apart, but that's it. You don't wanna unscrew anything or unbolt anything, because it is a really good microscope. It has a lot of places you can attach special features, but if you start taking it apart, it's a precision tool. It's really hard to get it back in place. The distance between the lenses is perfect. If you mess up that distance, you'll never be able to get a clear image again. And the last one is kind of covered up by the QR code, but always use lens cleaning cloth. And you have a little packet of lens cleaning cloth. And the reason is that this is 100% debris free. There's nothing, grit free. There's nothing on this that can scratch your microscope. Even something else that I keep in the lab, like they're called Chem wipes. They're thin, they feel a lot like lens cleaning tissue, but these are only 80% grit free. So there's still a chance that you can scratch your lenses. So when you clean the lenses, always use cleaning tissue. If you run out of this, you can go to an eyeglass store. You can order it online at Science Supplies, but always use that. The rest of the microscope you can wipe down with a Clorox wipe or anything else. It's safe for that. This QR code is actually a link to give you some more advice on things that you can do to clean your microscope and keep it in good shape. One other thing I might suggest is getting some rubbing alcohol. So 70% rubbing alcohol works, but you can damp that a little bit on a paper towel and wipe down your scope. It kills all the germs that are on it. It's also really good for cleaning tools in between uses so you don't contaminate anything. All right, let's get your lab out. I'll put this here. Like I said, I have everything in the box. Yours came in a big cardboard box. with an envelope full of all the supplies that you use for creating your own experiments. And, oh, forgetting something. So the first thing I'm going to do is I'm going to make sure that this is shut. So this is a lot of the, these are a lot of things that I like to tinker with. And here you can see I have my camera holder, phone holder. I have the microscope. Even in the box, it has the cover on it. Microscope slides, and that's a special camera. Cover that. Okay. So, first item on your list is your microscope. Put it down, pull the cover off. Just fold the cover and set it to the side. I'm going to move my box of goodies to the floor. So we can go through this. Right. When you're looking at your microscope, it has a cover on it right now. You'll pull this out and put the lenses on as we need them. And we'll walk through and talk about all of the items that are in here. So I'll set this to the side. You'll have a box of prepared slides. This is really important. Sometimes this can be confusing. I've seen a lot of people make this mistake before, but sometimes when you're talking, you're not paying attention. You might open this upside down. So I like to write the word top somewhere on it so it lets me know where the top is. Before you even open it, when you go to open it, this will flip up. That tells you the up decides the top. When you open it up, there's a piece of foam in. You leave the foam in. Here's a list of all the prepared slides that are in here. When you take these out and put them back, remember they're very thin. If they get broken, they can cut you. Always put them back in their appropriate slots because the numbers line up with it. So this is one of those things that as soon as you take a slide out, once you use it, you want to put it back unless you're going to be using it immediately again. Otherwise, it gets to be really messy. So they're prepared slides. Like I said, here's your smartphone holder that'll go right over the lens. Let's just go through the checklist. You have a second set of slides. I did the same thing here. I wrote top on it. So when you open it, you know it's up. The foam in here. This doesn't have a list. You probably could write a piece of paper in here, but this has 5 basic starter slides just for us to play with. And then it has 10 slides back here for you to experiment with. And I'll show you a way that you can make your own slides once you've gone past those 10 without spending a lot of money too. Right, next items, the lens set. You have 3 special lens pieces, and these are called Barlow lenses. So there's a tunnel right here. You should have a 10x lens. The X just means times magnified. So if you're looking through this and you've looked at paper, you'll see that right away it's magnified 10 times. Okay? It's designed to magnify 10 times. Some things you might notice when you put it up and you start looking at paper, how does the paper look? So if you're looking at text, what's it look like to you? So we have the tunnel chamber, 10x, and a 20x. So those are your 3 pieces, and we'll explain the Barlow lenses later when we need it. The phone mount, we already have. Make sure that you're checked out. The prepared slides, we already talked about. You have blank slides, we already talked about those. We have a box of slide covers, just like this, and we'll open it when we need it. They're very, very thin pieces of glass in there, so if you don't have to open it, always keep it shut. And those, well, we'll explain them later. You have a sheet of transparency paper. Where did I just put mine? I have a piece of thin cloth on the back of it, but transparency paper, and we'll use this to make slides for backup slides later. We'll also use it for other things. It's great for holding specimens. There are a couple images on there that I'll have you cut out. as we go through here. Ring stickers look like this. They're for making wells on slides, and I'll explain that later. You have 6 vials. A gum medium for making slides. You have sea salt for making water to grow your brine shrimp in. You have 2 containers of dyes and a container of yeast. Okay. So you actually have some sample specimens to grow right here. You have a hatchery, which is also a specimen dish, just like this. Lid comes off, so it's like a small petri dish. You can grow your brine shrimp in there or your yeast. You can also use it to capture specimens. You have lens tissues, tweezers or pickups or forceps, whatever you want to call them. Handling small things, or actually also handling things that you shouldn't be touching. You have a probe. You see it comes apart here. Goes back in. Oops, be careful when you put it back in so you don't poke yourself. A spatula. A small paintbrush for removing debris. You have a pipette, also known as an eyedropper. Same thing. The probe, you have a pencil that I pulled out earlier. You have a marker, and the marker comes with a ruler. Also, some little white tabs that you can put on your slides for marking them. Ruler already said. A few toothpicks that you can always replace. Just go to a dollar store and get a whole pack of 500 for like $1. You have a pencil sharpener. They'll be using also as a microtome, which slices samples. Clear nail polish, super dry, to help us preserve our specimens. Let's see, microscope cover, which we already talked about. Some clear tape on a roll. It's just simple Scotch tape, but it's crystal clear Scotch tape. So if you go to replace it, that's what you replace it with. A pack of batteries, 3 of them. Your notebook, which is your notebook you're flipping through now, / guide, and an owner's manual for the microscope that you have. So this will go through and remind you also some of the pieces or parts, how to use some of them in general with the microscope's for, how to fix the light if something goes wrong, how to put the lenses together, how to mount the smartphone, and also some of the explanations of what you can do with yeast, shrimp, and how to create your own specimens. So that's the set. You'll also have, of course, the STEM flyers and STEM bookmark, which is really important to have the bookmark because as we're flipping back and forth in between pages, you want to use that to mark where we're coming back to. That's the basic kit. There are a lot of other things that come with it, but these are the basic tools you're going to be using over and over again as you explore with your microscope. Okay. Next point is don't jump ahead. Make sure you go one step at a time because there are a lot of things that I'll talk about that I've referenced in the past that I really want you to pay attention to, and I'll try and point those out too. The first being your specimen collection. So when you get a hold of a specimen, when you collect a specimen, anytime that you're working with anything under the microscope, you want to make sure that you're handling it with care, number one, and you're taking care of the specimen itself, not just for your protection, but also for the specimen, because you may have a living organism. You might catch an ant or you might catch a bug. I just saw a bug walk through and I thought, oh, I should just stop and go grab it and look at it on the microscope, and I didn't. But if it's a live specimen, I'll show you later how you can catch a live specimen, put it under the microscope, keep it alive, and then let it free when you're done. Other times we'll be working with specimens that are dead. And unfortunately, most of the time with a microscope, you work with dead specimens. So whether it's a dead piece of tissue that you had to slice apart, even in the lab, it's a sample of blood that's not living anymore, or a slice of a tumor, or a cancer cell, or something you're looking underneath it. But you've caught it, you preserved it, and now it's dead. Sometimes you want to keep the organism in its environment, and that's what this is talking about. Is you can catch things from ponds, you can catch things from under your fridge, you can collect dust from under your bed, and you might actually see living organisms in there, which is freaky, but they're there. Heck, you could take a specimen, you could take a piece of tape and put it across the top of your eyebrow overnight, peel it off the next morning, and you'll probably find little tiny bugs there because most of us have face mites. It doesn't matter how clean you are or how dirty you are, they're just something that live on you. So when you're collecting specimens, you want to make sure you take care of them. The ones on the face, I don't think you want to put them back on, but there are a lot of places to look. Culturing a specimen is another important thing. So that's what we're going to do right now, because very soon we're going to be working with some moss, and we want that moss to be hydrated. So a lot of times you'll find something like a piece of moss like this, or you can take some lichen from a tree bark. And if it's dry outside, there's still living organisms on there, but sometimes they actually go into this stage called a cyst, where they put a protective ball around themselves. So when it's dry, they don't dry out and die. They just protect themselves. And then when water is re-exposed, then they can be rehydrated. They come out of their shell and they start living and acting again. They've always been alive, but they start actually being animated, crawling around, gathering food and doing what they do, replicating and stuff. So when you're using a specimen like this, you have to rehydrate it, and usually takes a while. It can take an hour, it can take 2 days, anywhere in between. So when I take a specimen and rehydrate it, I'll take a peek at it like an hour later, a couple hours later, the next day, and later that day. But you'll take your specimen cup. It's really important. If you're using something that's normally dry, like tree bark or moss that's on a rock, which by the way, on a rock is better than on a tree, you'll take your specimen, put it in your container, grab some clean water. If you get dirty water, then you're contaminating your specimen. You're not really going to be able to know if it came off the moss or if it came from your water. And you'll pour it. So I'll bring this a little bit closer to you. I pour it, whoops, I put a little too much on there. So I'm going to dump this off. Okay, perfect. So what I wanted here is I want just enough water to cover my specimen. I'm going to flip it over a little bit so I make sure it's well hydrated. And flip it back, and you can kind of see already some of the dirt is falling off. I'm kind of mushing it around. You can do this with a small Ziploc bag too, but the trick is that you want to make sure that there's still air exposed on the top of the moss. See, the top of the moss still has air, because these organisms want the air. They're in a dry environment because they need air to live. It's just like you. I put you underwater and I keep you under there, you're going to drown. So we want to keep them alive. I wouldn't put cold water on. I would put at least room temperature water, maybe a little warm, but not very warm. And I can already see it starting to spread out, if you can tell. Perfect. If I'm taking a sample of the lichen, I'll take my spatula, and what I'll do is I'll scrape some of the top off into the specimen cup. I'll break a piece of this off, and I'll put it in the specimen cup too, because then later, when this is soaked, the scrapings, I can spread those apart. I can look at them, but I can also take this piece and scrape some deeper tissue off, because now that it's wet, I can go a little bit deeper and see down in the wood, did something burrow into it, or is it all on the surface? That's culturing. We'll do the same thing with yeast when we get to it. Yeast is really fast. It takes only like 10 minutes to get it active and be able to use it again. So we're not going to cover that right now. But here's a QR code to some other things you can do with culturing. And then we just already walked through this. We'll actually do a whole lab on soil. So we'll do this with a little sample of dirt, because in one little teaspoon of dirt, there can be hundreds of thousands of little tiny organisms in it. Things that might be in this area of dirt outside of your house, but you go 10 feet away and none of them are there. So it's pretty incredible to just sample different areas of your environment. All right, let's get going on the main microscopy material. So always be inquisitive is this again. You ever wonder what makes your breath stink, or what makes armpits stink, or feet stink, or other body parts stink? it's maybe not what you think. It's not the sweat that does it. It's not because you wore the same socks 2 days in a row. It's what's in the socks. It's what's eating the sweat, and it's bacteria that does it. What's interesting is that for a long time, we scientists, medicine, thought that you could get sick from just the air that you breathe, not particles in the air, but the actual air itself, the stinkiness of the air. But the stinkiness of the air isn't what makes you sick. It's what makes the stink. It's the bacteria in it. Just as an example, in this situation, morning breath. You ever woke up and you're just like, oh, wow, breath is so bad. Other mornings, maybe not so bad. It actually is determined by how much you sleep with your mouth open, because the bacteria that's in your mouth all through the day, it's all there. But you usually have your mouth closed during the day, so it doesn't get a lot of air to actually metabolize things and build up the stink. But at night when you're snoring, whether you snore or not, with your mouth open, lots of air comes in. That bacteria is growing, it's releasing its chemicals into the air and the stink into the air. And there's your bad breath where it comes from. People, some scientists actually go a step beyond and get really kind of too inquisitive about things. But for instance, down here on the link, one scientist back in the 80s found out that all these people had bad breath, halitosis, which is bad breath all day long, not just when you wake up in the morning, but real stinky, like powerful, acidic smelling bad breath. And a lot of those people also develop stomach ulcers, which is when your stomach has like, it's kind of like if you get a cut, an opening, your stomach has this opening or pore on the inside that's an ulcer. It's irritated. It's not supposed to be there. And you have all this stomach acid that's eating at it. So these people that have really bad breath and they also got stomach ulcers, this doctor studied some of the chemicals in their stomach, took some samples of their stomach juices, and he found that there was bacteria living in it. Well, a lot of other scientists said, well, that's not possible because the stomach acid is so strong. There's no way that bacteria, this little single cell organism, can live in that. So he kept studying it, and he gathered these samples of bacteria, and this is where you should always draw the line. He ate the bacteria. So he was curious, and he said, well, if this bacteria is causing this bad breath and these stomach ulcers, if I eat the bacteria, then I should get the stomach ulcers and the bad breath. He was right. So he started getting really bad breath, very strong. I'm sure his partner didn't want to kiss him or anything. But then he started getting really upset stomachs and found out he was growing ulcers. So the bacteria he had eaten that was causing ulcers in other people was also doing the same thing in him. Well, on the plus side is that we know how to kill bacteria, right? The stomach ulcers that were a big problem, people were like, well, what do we do? Do we need to do surgery or do something different? People are drinking all these other chemicals that were doing nothing for them. What kills bacteria? If you go to the doctor and you have a bacterial infection or they suspect you of having some kind of bacteria in your body, they'll give you an antibiotic, right? So it kills bacteria, doesn't kill viruses, but it will kill bacteria. So you take this antibiotic and, a couple of weeks later, your halitosis is gone, your bad breath is gone, your stomach ulcers are getting better. It's like a miracle. And he found that about 90% of people that had this bad breath and stomach ulcer combination actually had this bacteria in them. So always be inquisitive, wonder about things. There's a little link down here. Oops. On the next page, I think. Yep. Oh, yeah, it's on one of the other pages. That actually is a link to our STEM program and microbes and how you can grow microbes at home just using slices of cheese. So maybe you're curious and you can do some different things. If you're wondering about what's growing on your face, take this piece of cheese, put it on your face, hold it there for a little bit, pull it off, put it in a Ziploc bag and see what grows on it. Wash your face afterwards, please. You can do that with anything. If you're wondering what's on the back of your hand, put the cheese on there. Wait a little bit, peel it off, put it in the bag and let it grow. But you can experiment in a lot of ways that don't require a lot of money or a lot of complex tools. You could take that slice of cheese and actually put it in the microscope and look at the microorganisms that are growing under it too. Right? So your next tool, first tool is your brain, second tool is your lab set, and your third tool is your notebook. And these are the three most important tools for a scientist. So your notebook should be a place for you to write down whatever you think. Some people call it a diary. Sometimes diaries have this negative idea about them that they're where your secrets are and about who you have a crush on, and who's cute in your class or whatever. But it doesn't have to be. A notebook can just be a place that you can write down ideas and thoughts and things you want to experiment with. Leonardo da Vinci had, when he died, he had 7,000 pages of notebook with over 7,000 drawings in the notebook of just random things. It wasn't just one thing. Whether you think of him as a scientist that explored and was curious, you're right. He did. He had a lot of questions and found a lot of answers to things. If you think of him as a painter, you think it's full of painting pictures. You're kind of right too. He had a lot of sketches of sculptures and artwork that he wanted to do. Like a giant horse they wanted to sculpt. There were pages of it, of how he was going to build this thing. Flying machines, catapults, tanks, just all kinds of really cool things. He had sketches and sketches and sketches. And what's really important is that he didn't build or draw, paint, whatever, a lot of those things, but because of his detailed notes and the ideas he had, the way he wrote it down, people today have actually built those things. So there's an exhibit traveling around the country of like the machines that Leonardo designed, but didn't even necessarily build, like his tank that he didn't build. So what's important about your notebook is it's a recipe book for you and a recipe book for anybody else that wants to build something from your ideas. So if you have this idea, write it down. Write down the things that you think are important about it. You ever woken up from a dream and you just have, this really cool idea, this brilliant idea, and you want to tell people, but you go downstairs, you brush your teeth, you grab a cup of water, and by the time you get to the table, you go, oh, I had the best dream. I can't remember what it was about. That's what a notebook's like. If you have an idea, write it down or you're going to forget about it. So an idea that's not written down is really worthless. So as you're coming up with things, whether you're working on moss in your notebook and then you think about something that has to do with cheese, write the cheese idea down too. Don't let it slip away. Your notebook's your best friend. So when I'm explaining a process or I'm showing you how to prep something, your notebook's another place that you want to write that. Make your own recipe book. So as you're writing, you can write down, here are the materials that we used. Materials. Here are the steps that we did. 1, 2, 3, 4. Write down, here are my ideas. Here are my observations. You can take it and actually set it up just like the scientific method. The other day I was noticing blankety blank blank blank, right? I wonder if, so as an example, the other day I was making Jell-O and I put grapes in it and pineapple, and what I observed is that it turned out soupy. It didn't work. So that was my observation, right? The next thing I want to do is build a question. So what's my question? Why did it get soupy? There's got to be something about it that made it soupy, right? My hypothesis, it's probably the grapes or the pineapple, because when I make Jell-O, my Jell-O is strong all the time if I just make plain Jell-O. But man, when I put these grapes and pineapples in it, it's soupy. It's got to be the grapes or pineapples. So my experiment, I'm going to build an experiment that says that it's one or the other. So in this situation, I might say it's the pineapple that's causing it. So I think the pineapple is making it soupy, not the grapes, just the pineapple. So I'll build an experiment that takes 3 cups of Jell-O. So I'll take 1 packet of Jell-O, I'll put out 3 cups, I'll put 1 cup and pour a third of the Jell-O in that with nothing else. 1 cup, I'll put a couple pieces of pineapple in, pour a third of the Jell-O over, and the last cup I'll put a few grapes in and pour the Jell-O over, right? I let it cool for 20 minutes or overnight, however long it takes for the type of Jell-O I'm using. And then the next day I observe. What do I notice? So I carried out my experiment. Now I need to collect my data. The regular Jell-O is perfect Jell-O. It's the way Jell-O is supposed to be. The grape Jell-O, the Jell-O part is just the way it's supposed to be. The grapes seem okay. The pineapple is different. What's different about the pineapple? Try the experiment at home. It's actually a true thing. If you like Jell-O, my mom always put fruit in her Jell-O, but never pineapple, and this is why. So my observation of what happens when the pineapple goes into the Jell-O is really important. Then I'm going to think, why is it? What is it about pineapple that makes it different than grapes or pear slices or cherries or whatever other things you put in it? Find it out. If you don't know, I'll give you a hint. I found this really cool thing on the internet. It's called Google. Yeah, it's spelled Google. Anything that you don't know, type it in. Type the actual question into Google. A lot of times you're going to find the answer. In fact, you'll probably find a lot more than just what you asked for. But it's the same thing with your slides. If you're looking at something under the slide, you can't figure out what it is, and this is the reason that I include the smartphone, use your smartphone or tablet or whatever that you can fit in the device, take a picture of it, and then take that picture to Google. You can actually do an image search on Google and isolate down to what it is that you're looking for, and then put it on Google. And a lot of times Google will tell you what you're looking at. So cool. If you didn't know that, now you do. Right? But keep notes. Write little Google ideas on the bottom of your notebook. Go back and reread your notes. Don't just write them down and walk away from it. Right? The next thing I really want you to do is think critically. And thinking critically is, man, it's probably the most important thing you could ever do in life in general, not just as a scientist or whatever, but just period. So the first keys here are actually wisdom, understanding. Look to understand, not just to know something. So I might know that red is a color that's on, whatever kind of car, but I really want to understand why they put red on a car. So why would they put red on so many cars? Well, probably because it's a popular color. It sells most cars, right? But understand why you do what you do, not just doing what you do or knowing to know. Right? Have an open mind. Be creative. Think of other things that are connected. One of the coolest things I found about science is that everything's connected, really. If you learn certain rules in biology, those rules apply in chemistry and physics. So a lot of principles, like the way that electricity and water move, it's connected. Have an open mind and look for those kind of connections. You might get an answer for a question you have by looking at a different area completely. Sometimes take a step back away from things. At one point of time, we didn't understand the microscopic world. Now we do, and it's really cool because you're going to learn a lot about a lot of things, or look at a lot of things a lot of people have never even seen before. Like I said before, we didn't understand disease before microscopes. We understood that people got sick, and there might be some ways to treat them. Otherwise, they died. We got that, but we really didn't understand that germs were really the cause of most diseases that people worry about. Just as an example, there's a QR code to John Snow. He was probably really the first epidemiologist, the first person to study a disease that spread across the country. And John Snow, back at the time, it was called miasma. If you breathe stinky air, you could get sick. That's miasma. They didn't know it was something like living in the air. They just thought it was stink. So in London, where John Snow was at, he was starting to do a study of where people were dying from this cholera sickness. So cholera, they would get really sick. They'd have diarrhea, vomiting. They get so dehydrated, they would actually die really bad. The cure is clean water. If you didn't know, it's actually the best cure. I mean, you could take antibiotics, but just getting clean water, you'll get over it. But they didn't know that back at the time. And so John Snow looked at all the places on a map that people were dying. He was keeping track. And he noticed that were like big red areas where he was making these dots that people were getting sick a lot. And he said, what's the common cause? If it's in the air, what is it about this air? Where's the air coming from? So he looked around and said, well, here's where people shop, but not all these people went to the same shopping area. Here's where people go to the church, but this is not always the same church that these people are going to. Here are the other places people socialize. Here are the bars or pubs that they'd go to. Here are the different things that people would go to, but he wasn't noticing any real connections that like all these sick people went to the same place, except a well. He found that back in the day, they didn't have a faucet that you just went to and turn it on. They had to actually go with a bucket to a well and use a handle and pump water, bring it up from underground, bring it out and put it in your bucket and then carry it home. And he found that one well was central to most of all of these sicknesses. And so what he did is he went to the church, and back in the day, the church and science didn't always get along, but he went to the church and said, I really believe that this is the cause. And so the church supported his idea, and they took the handle off the pump. So now people had to go to other places to get their water. And what he found is that when people were going to other wells, the sickness was going down. So the well was the cause. He observes things. He built a question. He built a hypothesis and said, it's probably this well. Built an experiment. Let's take the pump handle off. And then he observed the outcome. People were getting better because they were going to clean water, drinking it instead of drinking from this well that was infected with cholera. And when the microscope came about, they studied that water, and that's what cholera looked like. So cholera is a bacteria, Vibrio cholera. And we understand diseases. So because he was inquisitive. He had an open mind. He was thinking out of the box. Like I said, the church and science didn't always get along. He was using a scientific method to solve a problem that a lot of people thought was, the hand of God or in the air, or just, things that actually weren't true at the time. So detailed observations, measurements of little things, keeping your notebook is really important. And when it comes to the little things, this is really cool, and I had to share it, but there's actually a website that helps you to get an idea of how big things are. So let me go down and open that for you. And I've included the QR link too, but it's Scale of the Universe. You go to that, you can click on your language and then hit start. Oops, do it on here. There it goes. So you can see the size of you, size of a basketball. We can slide the scale and look in smaller, or you can just scroll. You can see the size of the penny compared to a matchstick or an earthworm. Microwave wavelengths, pretty awesome. Coffee bean compared to an ant. You can just keep going deeper and deeper and deeper. So there's an ant, and you can see the size of bacteria. Here's a dust mite. Here's an amoeba, so a germ. What I thought was really cool is that when I was in college, I saw a picture, and I wish I had or had the rights to it, but it was a picture of a mite and the bacteria on a mite, and then a virus on the bacteria. So the person that took this picture had actually got this mite, and they were dying of this disease that was caused by a bacteria, and the bacteria actually had a virus on top of it. So they kept zooming in and zooming in with microscopes to actually see. So here you have, there's a dust mite, and there's human hair. You can see there's the ovum. That's a female egg from a person, close to the width of a human hair. Silk fiber, we'll actually be taking a peek at that. A mist droplet and a silk fiber. A silk fiber from the silkworm is actually 1/2500th, sorry, yeah, 1/2500th of a millimeter. Super, super tiny. And you see a water droplet is about the same width. There's a white blood cell that protects your body. Your chromosomes, you can see there's your chromosome, what makes you up. And then there's E. coli right there. It's actually smaller. Keep going down. You can see different colors of lights, wavelengths. Red, violet. The biggest virus, there's a bacteria virus. Keep going. There's HIV, so super teeny tiny. Think of all the things we've already passed already. Just keep going and going and going, all the way down to the building blocks of your cells. Here's what makes the wall of your cell. Here's a particle of sugar. Right there, just one. There's an atom, and then even particles of atoms. So super tiny. But it's a really cool website. If you get a chance to explore it, totally recommend it. And why not just look around a little bit and say what's in the human body? Because I think we passed several cells that are in the human body, like blood cells, immune cells, and we'll talk about those later. But think about the size. Winding down to this section, complete history of microscopes. Maybe not the complete history, but kind of the cool history. So microscopes are really not that old, but when you think of man looking at things, we've been magnifying things probably for as long as human history has been written, where man would just take a piece of broken rock and look through it, because some rocks actually are clear and they can reflect light really well, refract light, so we can magnify. I'm sure there was probably a caveman sitting around somewhere that actually took a piece of quartz and held it long enough in the light and saw that it had a small beam and probably accidentally created fire. But that's what I like to imagine. You never know how old magnification is, but to magnify is only one part of looking through a microscope. Magnifying is just making something bigger visually. It's not actually making it clearer necessarily, and we'll talk about the differences. So some of the first microscopists were actually people that would take a piece of glass and polish it so well with really fine claws for a long time until they had a really clear image, not just magnifying, but actually resolving. So seeing the difference between two items that are magnified, that's resolution. The first compound microscope was in 1595. So Compound means that there were two lenses. They figured if you take one lens and you add another one to it, you can amplify that. And that's exactly what we do. When we look at our microscope, you'll have a lens here called an ocular that you look in. You also have an objective lens down here that magnifies it. So if this is 10 times multiplication or magnification, and this is 10 times, you've actually got 100 times magnified by having two lenses appropriately spaced. In 1665, a doctor, Robert Hooke, actually looked at cells. He looked at a piece of cork and identified the little tiny structures. We'll add this to our book, or you can Google it because there's a really cool picture of it. But he saw that cells were these blocks, almost like Legos stacked together on a single slice of cork. And he called them cells because they look like cells in a monastery, like where the monks sleep, or I guess you could think of a prison where the cells are lined up. So he called them cells. And one of my favorite pieces of microscope history is actually Anton van Leeuwenhoek, that he was actually a seamstress. He made curtains, and he made expensive curtains out of special fabrics from all over the world. And so he built lenses and polished them so well, better than anybody had before, so that he could see the fibers on the fabrics and know that if it was real or fake. So if you're trying to find something from, we'll say, Asia, that had real fine silkworm woven patterns to it, you want to make sure it's the real thing. So that's what he did. But he got so good at it, he was seeing a lot of things that people hadn't seen before. So he built this little tiny microscope, and we can build one right now. If you take a note card, this in your first envelope. So sorry, your envelope looks like this. And I'll say 101, whoops, up in the top corner. And it has all the materials like this card in it. And in the card, if you take your pencil, this, kind of poke it in and twist. Right, you get a nice round circle. I like to take, where to go? I'll grab a new one, a ring. I can take this ring, I'll take a green one. Put it around that circle, just like this. I'm actually going to leave those sticking up a little bit. But now I'll take a piece of tape. And with the tape, find the edge. Always the worst thing about tape. And I think it's the only thing I don't like about Christmas is the tape with the wrapping. I like wrapping, but I don't like getting the tape off. So let's get the edge of this tape. We just need a little tiny piece. Wow, it's being really difficult. Here we go. Got it. So a little tiny piece. And I'm going to take my probe and just pop a hole in it to cut it. And not on the side with the ring, but the opposite side. Put tape right here. I have some water in the cup. I'll take my eyedropper, pipette, and I just want to put enough water. Drop. Perfect. Man, that's awesome. Just enough water so it sits inside that well. So this little ring we call a well. I can see my little drop of water. And now, if I bring it down, I have a small magnifying glass. I can actually look at the magnification of the print. So it's really cool like that. And the reason it does that is because of the roundness of the water and some of the properties of water. But van Leeuwenhoek, when he built this, he polished, instead of using a drop of water, he polished a lens, real fine lens, put it in there, and then he had a small stand that he would dip into water, like a toothpick that was inserted, and he would hold it. I'm not going to be able to do this quite right without dripping the water, I'm sure. Oh, wow, it stayed in there. Great. But he would hold it with the drop of water, his lens over here, and he would hold it up to a light. The picture I remember seeing is he would hold it up to the sun. Not a great idea. But he'd hold up to the light, and he could actually see, because of that, magnification, little tiny things moving inside the water. And so he drew them and he called them animalcules. And those were the first tiny microorganisms that were recorded. So, and he was just a seamstress. He made curtains, but he noticed living things. And so he started looking at everything under the microscope. He looked at scrapings in his mouth. He looked at spit. He looked at drops of blood. He looked at everything he possibly could. So, and now he's famous forever for it because he did that. And he was not a scientist. He was a seamstress. Other types of scopes. Nowadays, we have things that don't use necessarily light. They use electrons, which are particles of, well, electricity that go through the image. So if we take a cell now, we took that same cork cell, we can actually look at it through an electron microscope and see a lot of the details inside one individual cell. Okay, Robert Hooke didn't see all that detail. He just saw the frame of the cell and sometimes a nucleus. The nucleus is a little dot in the middle like that. Right? We have special scanning electron microscopes that we can get 3D images, really good 3D images of here's a cork cell. Here, if you look down really close to individual pieces of cork. So it's amazing the things we can do with microscopes now and all the fields that you can use them in. In fact, we're going to go through a lot of these fields in the next 2 sections. We'll talk a lot about medicine, of course. I'm a biomedical scientist. That's my background. We'll talk about food and how microscopes are used in food treatment, and we'll do some experiments. We'll talk about material research like textile industry. We'll talk about metals. We'll look at some money under the microscope. We'll talk about geology and rocks. In fact, one of the other sections, you'll have a diamond in the set that you'll get to look at, a real diamond. It's a raw diamond, and you'll find that and you look at it and analyze it with a microscope. So gemologists, people that study diamonds and rubies and sapphires and things, they use microscopes every day, magnifying glasses and microscopes. In geology, we want to understand the rocks and the things like microfossils that are in them. Forensics. Forensics is interesting because it actually takes almost every field, uses a microscope, or every field of science and does something with it. So we'll talk about forensics and how crime investigations use microscopes, and you've probably seen this on TV. Archaeology, so how can we date something using a microscope? How can we know that fabrics are from a certain period of time or a certain country? And we'll talk about microbiology, so the germs and microbes and yeast and all the cool things that you can look at. Micro meaning small and biology meaning living organisms or the study of living things. Right? There are a lot of really cool pictures you can look at with this QR code. And then the last section, let's talk about how vision works and how your eyeball works. So vision is really cool and interesting. If it weren't for vision, you wouldn't be able to use a microscope, right? Microscopes and the eyeball work differently though, but they both work with light, right? So light, if you didn't know, is wavelengths. So it's wavelengths that are moving through the air. They're coming in and penetrating your eyes and you can see it, right? So this movement of these wavelengths, if it's coming off as white, is actually carrying all of the wavelengths of the other colors. So when it hits something, 2 things can typically happen. Either light is reflected or bounced off of that thing. Like if I'm looking at a tree in the distance, and when white light hits it from the sun, as it hits the tree will soak in all the colors except one. There's one color that's reflected or bounced off, and that's the color we see when we look at trees, which would be what? Did you think brown? Okay, so the bark would be brown, right? Most people think green, like the leaves, right? So when trees make light, what's interesting is that the color they don't like is green, and we always think of green when we're thinking of like chlorophyll or chloroplast or the parts of leaves that make sugars. So reflection is when the light bounces off something and then goes into your eye, and your eye detects that color. It's the color that's rejected by that object. Refraction is different. Refraction is the bending of the light, not reflecting or bouncing off, but it's the bending of the light as it passes through something. So when I'm looking at a microscope slide, this, if I'm looking at the slide, you can see me right through the slide, right? I can hold it up here and you can see right through it. Light passes straight through. Even though it looks like it's perfect, it's not. So it's refracted or bent slightly, maybe a lot, depending on the thickness of the glass, the shape of the glass, depending on the water, everything. Let me give you an idea. So I'm actually looking at this example right here on my table. So there's a glass, right? If I take a pencil, and you have a pencil, you have water on the house, you also have a glass. But if I take the pencil and I stick it in the water, notice that change kind of bends. Right? Whoops, rolled around. So now I'm not getting a perfect bend. Let's do it like this. Get a bigger glass. Oh yeah, lots of water. Okay, so the bend. Noticing the bend right here? The bigger the glass, the more water, depending on the shape of the glass, you can get greater effects, but like that. And I know you've seen this in the swimming pools where you look down at a friend from the top and the top of the body is really long and then the legs are real tiny because of the refraction, the bending of light as it goes through this water. So it shifts it off so that it looks slightly different, right? That's refraction, that's bending. Water does it, plastic does it, rocks will do it. So a clear rock, as light goes through it, we'll actually split it and we'll test that in one of the other sections. Right? That's refraction. Sometimes some objects, as the light passes through, will only let certain wavelengths of light go through. We'll test that too. Your eye uses refraction. So here's my eye. Whoop. There it is. When light passes through the front, it's kind of partially taken apart. When light passes through the front, there's a series of things, we'll call them. Tissues, fluids, different things that refract or bend the light. So I don't want to say straight water, it's not glass or plastic like this, but you have organic materials that light passes through that bend the light as it passes. The white part of your eye actually reflects light. So the white part of your eye, the light hits it and it bounces back. What wavelengths come back? What do you see? You see white, so all the wavelengths are coming back to you. The white part of your eye is great because it blocks out all of the light and only lets light pass through the center part of the eye here. So all this white bounces all the light off. If I were to take this apart, I can look at where it's going. So the first thing that light passes through is called your cornea. And your cornea actually does a lot of bending. About 2/3 of all of the light refraction happens right at the beginning in the cornea. We don't give it a lot of credit for bending light, magnifying light, changing light, because it's not very manipulative. We can't manipulate it very well, right? Well, we can. As you get older, and if I weren't wearing my glasses, you'd see me manipulating my cornea a lot because we do this. We squeeze, and what we're doing is we're trying to bend the cornea, squish it down so that it bends light differently. That's what your cornea does, bends light. Also protects the front of your eye. So if you get dust or scratches on this, it's just like dust and scratches on your lens and your microscope. It's bad news. The second thing light passes through, oops, is your pupil. Right here. This part's called the iris. The iris is adjustable. Some microscopes actually have something called a diaphragm iris that you can adjust the amount of light coming through into a pinpoint or wide open. On bright sunny days, you've probably seen your pupils get really small to only let a little amount of light through. On dark days, way open to let lots of light through. So you get a better image of what's coming through. Right? So once the light's gone through the pupil, then it goes through the lens. And the lens is what we usually give credit for bending light. About 1/3 of the bending or manipulation of the light is from the lens. We give it credit because when the lens is in place, I keep breaking it, but you can see that. There we go. When the lens is in place, there are little tiny muscles right here to the sides of the lens. Let's put that again. That's why. I'm not going to be able to get it back in. I'm just going to keep fighting. Oh, there it goes. So you can see the little tiny muscles to the sides, kind of brown in this picture. Right? But those muscles grab a hold of the lens and they pull it. When they pull it, it flattens the lens and you see things in the distance really well. When they let go of it, boom, the lens is round, so it bounces back into a more round shape and you see things closer normally. When babies are first born, they got really round, squishy lenses, and they see things up close really well. That's why they want to see things really close. You get really close up on a baby's face and they freak out because now you're huge and clear, right? As we get older, we can't stretch that, just like the rest of us. We don't stretch very well, so you can't stretch it and the lens stays flat. That means that as you get older, you see things in the distance really well. It's called presbyopia. Myopia is seeing things up really well. Presbyopia is seeing things way out in the future, or in the away from you. My grandfather always joked because the older he got, the further he'd have to hold the newspaper to see it clearly, tilting like this. And he said his vision wasn't getting any worse. It's just that his arms weren't getting long enough. Right? So now the light goes through the lens, gets bent or manipulated. This is not solid in your eye. It's actually jelly-like. It's called the vitreous humor. There's an aqueous or watery liquid right here behind the cornea before the lens, and there's a vitreous humor behind it. This fluid also bends the light, and it's actually really cloudy. So if you were looking at it, or if you've done one of our STEM dissections when we dissect the eyeball, if you haven't, you should get it. It's really cool. We'll send it right to your house. But when you dissect it open, that jelly is kind of cloudy. So your vision is really kind of cloudy, and your brain fixes all that. But the light has to go through here, and then finally hit the receptors on the very back, right? And pointing at the wrong place, because this actually has no receptors. This is a blind spot in your eye. Google the blind spot in the eye, and it's a really good experiment that shows you how to use that blind spot. But as light comes through, remember, you're taking light from all of your world like this coming in, so light will come through and actually come over like this in a hidden area called the fovea. It has a lot of color receptors, so you see color really well. And all these structures we talked about coming up here are meant to focus the light on that area, right? So you get a good color image. You see things bright and colorful. If it's not on this image, or sorry, this little yellow spot, all of the rest of this area over here and over here are full of other structures that detect black and white. So your color vision is really only right here. At night, this spot is pretty worthless. So at night, you want to see things in black and white. Your vision is actually better to the sides. Try this sometime. I have a hacky sack at home. I should probably bring him to the lab, but it has 4 colors on it. And I do this sometimes with my friends just to show this one cool. Look straight ahead and just randomly spin the hacky sack and bring the color or the ball forward. Keep your eyes straight. And when you see the ball, try and name the color you see. So as you bring it up, it's really hard to do because when light enters your eye from the side, it goes over and hits one of those black and white receptors, not color. When you finally bring it far enough up that the light is hitting color, then you know it's colorful. So a lot of times, unless people cheat and they turn their eye to the side, when they're looking like this, they can't see the color until you get way up here. It's because of that colored area. So if you want to see something really well at night, don't squint and focus right on it. Kind of squint and focus just to the side of it, just so that that image goes over to the black and white area. It's what sharpshooters and bow hunter people and stuff like that, when they have to focus on something at night or in the dark, that's what they do. I don't know why you're bow hunting at night, but that's how the eye works. The one thing that makes the eye different from the microscope, which we're going to talk about in just a second, is that the eye has all the structures of a microscope for magnifying, but your eye has a vision receptor back here that helps you send a signal to your brain and actually understand what you're looking at. A microscope doesn't understand anything. A microscope is just a tool to get the light magnified coming to you. So there's a difference. Right? You can actually replicate the lens in your eye with a Ziploc bag. So if you grab your envelope, get another little Ziploc bag. Pour. Yeah, just reuse the water. There's water in it. Like this. And here's where it gets a little messy sometimes. See if you can keep all the water in. Perfect. Now you have this nice round lens. And you can roll it over the words and you can see they're magnifying. So I can magnify it. Bring it close, it's about the same. The further away I get, the more it's magnified. Another fun thing to do is squeeze it. Whoa. So if I squeeze like this, it changes the magnification without changing the distance. because the rounder it is, the more it magnifies. The flatter it is, the less it magnifies. So I can see things better with that roundness, but the more flat, I don't. It's just like looking through the side of a cup and magnifying something compared to looking down through the bottom that's flat. So you have a flat top and a flat bottom, it's not going to magnify. I can read everything, but it doesn't change the magnification. But if I read at an angle, this, it does make a difference. So you can make a water lens. If you're ever looking and squinting at something and you can't read it very well, put some water in a Ziploc bag and there you go. All right, let's break down the microscope. Not like break it, break down. Let's talk about the different parts of the microscope. Okay, when you're looking at your microscope, number one, two hands all the time, right? As we go through this, it's called a compound microscope because it has two lenses. The first set of lenses right here, we're going to put this, we're going to pull this out. Borrow the lens out, slide it into the tube. Second one you'll grab is the 10x. WF 10x means wide field. We'll explain field of view later, but it just means that you have a more broad picture, right? So this first thing is called the ocular lens or the eyepiece lens, and it magnifies 10 times. If you pull that back out and you look down through, you can see it's automatically magnified. Also note on the paper, also notice what happens to the print as you're looking at it. All right, as we come down here, we have the revolving nose piece. It's the thing that rotates like that. Maybe I'll hold it like this. There we go. I know I'm not holding with two hands. Feel a little unsafe, but revolving nose piece revolves. Hear it click, click. Now I know that this lens, which is called an objective, is in place. Rotate it, click. Now I know this objective is in place. Click. One of the main problems when you're looking at a microscope and the image isn't coming through or it's dark is that you didn't click the objective into place. So we have 10 times magnification. Coming down here to the revolving nose piece, we have different magnifications down here. So the one you'll hear me use the most is scanning. It has a red line on it. It says 4x on the type. We call it the low power or low power magnification. It's called the scanning because when you're scanning your sample, that's the one you always want it on. Right? When you're ready, you'll click to a higher power, which will be the yellow. That one says 10x on the side. Double check. So with the first one, I'm going to go back 10 times and 4 times magnification. That's 10 times 40, or sorry, 40 times magnified, total magnification. If you go to this, I have 10 here, 10 here, 10 times 100 is 100 times magnified. The last one is 40x, so I have what? 10 times here, 40 times here, I have 400 times magnification. So that's my total magnification. When I look at something under this, I've magnified it 400 times. You can switch this out for the 20 lens that you have too. So if that was 20 here and 40 here, it's what? 800. Yep, so 800 times magnified. Pretty cool. Right, the next thing we're looking at, the illuminators. You have a top illuminator. And, oh no, I did that. Might have to replace those. So another thing, take the lens out before you flip it over. I'm going to put some fresh batteries in so we can make the illuminators. Skip this part right now. Let's skip that part. So the illuminator, when you flip the switch on the back, number one is the top, right here. Number two is the bottom, and you can figure that out. You just get used to which way you're flipping. Right, so the illuminator is just the light source, fancy word. Okay. Going down a little bit more, this black surface here that you put your specimen on, your slides on, is called the stage. It's like an actor. You put your actor on the stage to observe it. These are called spring clips. So a lot of times when I'm working, I'll be holding my specimen, and I'll have my thumb here, and I'll spring clip, so I can pull the specimen back. And there's one on each side. You don't always have to use it. You just flip to the side if you don't need to use it. Right? That little glass thing down the bottom, let's turn to the low scanning. See that glass circle? That's your condenser. So it's a small lens that takes the light and condenses it down to a fine point or a smaller circle so that you have a lot of light source coming into your specimen to make it the brightest you can make it. Right? Underneath the stage, say, normally we call this the diaphragm right here. So the diaphragm is the round opening. Some microscopes have different size openings, or they have a special lever that lets you adjust it. Yours has an open diaphragm and then filter colors. And we'll talk about the filter colors. So you would just rotate it around to get the different filters. Right? You want to keep it open most of the time, unless you have a reason to turn a filter on. Next thing you want to look at is the arm where you hold it. Here are the control knobs. So the wide one here is the coarse adjustment knob. The little one is the fine. So if I take the wide one, I move it, you can see I get a lot of movement out of my stage, going down all the way right now. Bring it all the way back up, almost, and then take this fine adjustment, little turns, make small movements. So when I'm just scanning, looking around for something, I'll use the coarse. Coarse is only used with this red lined lens. That's the only one. You never use coarse with the other ones because coarse makes too much movement and you can actually break your slide. Because look at these. Look how low that sits, very close. If I adjust my stage fast with a glass slide on, I can break the slide. Don't use coarse when you're using the longer objectives, the higher powers. Okay. We talked about condenser, diaphragm, and the filters. The bottom illuminator we talked about. The rheostat is this knob on the side that adjusts the brightness. So you can turn on the light. Don't always need the brightest light you could possibly get. Sometimes you can dim it down and get a better picture. Right? And then the power illuminator switch, and then of course the base on the bottom where you're sitting it. Always sit your microscope on a flat surface. Don't rest it on your leg to look at things. It's awkward and it's not stable. Okay. And then down the bottom, I included a couple of QR codes. One has a practice lab that you can do virtually online to practice tuning and playing with knobs. And the other one is the actual virtual microscope, so you can tune or you can practice tuning and playing with things. So the lab tells you how to use the virtual microscope. And so if you're on the run, I think it works on mobile devices too. So you can play with it on a tablet while you're riding in the car or whatever, if you're just wanting to get good at it. Because working with a microscope is a lot like working with an Etch-a-Sketch. I know you've all played with it. It's kind of awkward and weird to use. So let's take some time and play with that awkward, weird to use. So the next page, your first look, right? So what handed are you? Are you right or left-handed? The nice thing is with the microscope, it doesn't matter because the control knobs are on both sides. They both do exactly the same thing. Everything's centered. It's monocular, which means it has one eyepiece on it, which I should probably put back in while I'm thinking about it. As I drop it again. Great job, Shannon. So as I'm looking through, what eye are you? So this is the tricky one. Here's how you can figure out what eyedness you are. Take your hand, put it up like this, right? And focus on something. I'm actually focusing on the camera, just like this, right? Both eyes open, close an eye. Can you still see the camera? Can you see the thing you're looking at on the wall? Can you see my hand or my circle if that's what you're focusing on? If you can still see it, click the other eye. Can you still see it, or did it move? So if it moved, that's in this example, my left eye is closed, that means I'm left-eyed. I have left eye dominance. So whichever one moved the image, that's your powerful eye, right? So my eyes, and this is actually how my glasses are too, if I take my glasses off, one of my eyes makes me squint. If I actually close my right eye, I can see things more clearly with my left. My left is my dominant eye because when I looked through, that object moved. My left eye is sending the main vision to my brain, and the right eye is just adding supplemental information. So I'm right-handed, but I'm left-eyed, which is good because when I draw with my right hand. I look into my scope with my left eye primarily, and I draw. Left eye, draw. Left eye, draw. It's up to you. If you wear glasses, you don't have to keep your glasses on when you're looking through, unless you have stigmatism that might affect it. But typically you can take your glasses off because you're adjusting the clarity of your focus on the microscope. So try it both ways, try it with your glasses and without. So your first use, flat surface, turn on the light. I actually have another one set up already right here. I know how the light works in this one. So I'll bring this over just a little bit more. Okay, flat surface, turn on the light. We'll use the bottom light. So which number are we looking at? Number 2. Yep. Oh, sorry, this one's backwards. Number 1. So bottom light's lit up. My stage is ready. Rotate the nose piece so I have scanning on. Remember, scanning is the shortest. Right now I have the longest. That's not what I want. There we go. Shortest one with the red line. I want to bring my scope all the way up. Okay. So I want this, and I want to bring it all the way up. Now I'm going to take my slide, whatever it is. I'm actually just going to use this for now. I'm going to slide this in. So here's your first use of a bookmark. And I'm going to take a peek in. Slide it down, slide it down. Oh, it's too dark. So since it's too dark, that means that this material is too thick. I'm going to flip my light to the overhead light. And I'm going to slide it down until I can see that paper. And now I can actually see the blue-green of the paper. I'm going to adjust the focus. This one I don't have to show you on the screen right now because it's pretty easy to find. So I'll adjust the focus so that it's clear to me. Cool. That's coarse. Now I'll adjust the fine focus up and down, up and down. So it's nice and clear. At that point, I can flip to the next objective, which is the yellow stripe, 10 times magnified. And the only knob I'll touch this time is the fine focus, the little one. And I just refocus. Now I see the same image, but I see it blown up. I go one more. Yeah, and that's too close. Right? To take everything apart, I just go back to my scanning, my red one, pull my specimen out, and I'm ready to go to the next. Okay. When you clean your microscope, something that's really important, never ever use anything except lens cleaning tissue. So if you see some dirt or things look cloudy, you can clean your objective lens, your ocular lens, you can clean your objective lens, you can clean your condenser lens that's down here on the stage, that little light here. You can clean the lights themselves and then try it again. But always use lens cleaning tissue. The rest of the microscope, you can use like a Lysol wipe or something else. But in the beginning, you always, or for the glass, you always want to use lens cleaning, always. Right? So let's look at our first samples under here. I'm going to go through these same steps again, but I'm going to use a sample. What's my specimen? Doesn't have to be something on a slide. It can be something really simple like, well, in this example, we're going to look at skin. So you can see a picture of the skin here. If you were to take a slice of skin, you can see the different layers. You see the surface, you see hairs popping out. But what's the skin look like under a microscope? Flat surface. I'm going to drop this to the lowest because I just want to stick my fingers underneath. I'm going to turn my top light on. There we go. And then I'm going to stick my fingers under. So here is where you can use your smartphone if you want to. So here's your smartphone stand. Pop it on. You have to have a lens in, whether it's the 20 or the 10, it doesn't matter. You have to have that lens in. I hang this. This part right here, I'm going to loosen. Not completely take off, just loosen so I can slide it up and down. There's still slide, but not too loose. I'll put this on. Oops, drop down. This takes a little practice. This is where your parents might be handy, but I'm going to bring this over. And I'm going to switch. So now you can see my phone. In this situation, I have a Samsung phone that I can plug into my laptop and it sends the signal through. There's my lab. Woo! You see everything. And I'm also going to wipe this with lens paper. I don't think anybody really wipes their cell phone camera with lens paper, but when I put this on, the first thing you want to do is you're going to look through the scope. So we come on. There we go. You're going to make sure you see a nice bright light. I mean, don't stare real hard at it, but you see a nice bright light. Okay. The second thing you're going to do, put something underneath here. I'm just going to put this back under again like I did before. I'll look at it. I flip the light to the top. I'm looking at it. Okay, I made it clear with my eyes, so I can see in here what's going on. Now, since I know that the lens is set up, I'm going to bring my camera over. I'm going to open these just a little bit. Putting one side in, the other side in, not hitting my power buttons. And then you can see centering. I'll slide it up. Kind of guesstimate. Okay, there we go. I just saw it. Here's my camera. There it is. It gets tricky. I said you might want to have a parent help you. Oh, look at that. There's my image. Right. That's a pretty good spot. Oh, I missed it. So with one hand, I'm holding my phone. The other one, I'm tightening the screw on the bottom. Is it perfect? Nope. Does it have to be? Nope. So for you, I'm sending the image through my computer. You don't need to do that. If you want to hook your smartphone up to it, what's beautiful, look at that, I just slid it up a little bit. Slide the phone down a little bit. Oh, I let go. Oh, now I'm way off. When you hook up your smartphone, it's great because you don't have to look down into the microscope all the time. Once you get this all set up, a little bit of shadow, which means I'm off to the side. Once it's all set up, you can take pictures on your smartphone and send it to your computer. You don't have to have it connected to the computer all the time. You just put the smartphone on, you can take a picture. There's me fine tuning. Something that's good to do is shut off the autofocus on your phone too. All right, so I have a little bit of shadow on the side, but otherwise, I'm just going to keep messing with it until I mess it all up. Otherwise, it's, yep, I did it. It's a pretty good image. There we go. There we go. You can see the colors. Perfect. Smartphone's ready. Now, I can lower my stage all the way down. I'm lowering the stage with this coarse adjustment knob. And now I'm going to check out my finger. Sweet. Don't push down. Don't push on the stage with your finger. Just rest your finger. I'll use my pinky. Ugh. Someone needs a manicure. Look at that. Terrible. Oh yeah. Pretty sweet. See the ridges and the lines on my finger? I'm going to take a note because that's what I do, because I love my notebook. So I've already done it here, but I just made a circle and I kind of made the pattern that I saw. I saw a nice even pattern. So those little swirls or lines on my fingerprint. Pretty cool. And the way that I had my finger was pointing like this. So you don't have to be a great artist, right? Just make a note. That's my pinky. That's what the ridges look like. And they kind of dip down. So I kind of shadowed that in a little bit. Now I'll go back and look again. Oh yeah, still looks the same. Crazy. Those are called papilla. We'll actually talk about them later. All right, now I want to see what the top of my finger looks like. I'm not going to be able to get my pinky in there, so I'm just going to have to skip to a different finger. Oh yeah. So I saw those ridges on the back of my finger, or on the front of my finger. That's not a ridge. Huh. That's a wrinkle. It's different. You can see kind of dry skin. Ooh, embarrassing. A little dry skin there. Oh, nasty. What's that? That's the edge of my nail. So that's where my skin ends, and there's fingernail. I'm fine tuning, fine focusing. Look at that. I have a scratch on my fingernail. All fingernail. Look at the change. What's really interesting is that your skin here is the same as your fingernail. There's my nail bed. There's the edge of the cuticle. That's pretty cool. So maybe I would take and draw a picture of that too, because I can see that wrinkled skin right there that's kind of folded over, and some of it's peeling off. It looks more thick and dense. And then there's the hard fingernail, right? So it's made out of the same thing. Our skin's made out of something called soft keratin, which is connective material inside the cell that makes it kind of tough, where our fingernail is made of hard keratin. The same with our hair. It's made of hard keratin, and we'll talk about those later. So you could look at your skin. You don't need a slide. You don't need a fancy setup to be able to look at something kind of cool. I mean, you can look at an insect leg. In fact, we'll be looking at an insect leg pretty soon. But don't forget in your notebook, write down what you see. What strange things did you see that you didn't expect? Did you expect to see a scratch on the surface of your fingernail? You know, look under your fingernail bed. So way down here at the end, see if you can see some dirt and nasty, grungy stuff. If you can find a hair on your finger, look at that. I actually have a scar on my pinky. Well, yeah, it's going to be really tough to look at. Let's see if we can check it out. No, there's the edge of my finger. That's the scar right there. You can see the difference in the skin. It's more pink on one side and it's more clear on the other, where the scar's at, because the scar is not fresh, living, like vital tissue. It's old and it's packed with keratin because it's been damaged. So you can check out those things, look them up. Okay. A lot of questions you can ask. Next, let's look at some pictures. We already started doing this. Heck, I'm just going to take, you can take this with your notebook, but I'm going to take this page out. I'm going to look at the tiger. So tigers are what? Orange, black, and white. That's all I see on there, right? Orange, black, and white. There's no greens or yellows or blues on a tiger. So I'll slide it in here, make sure my stage is ready. Slide it in. I'm going to slide it until I see the edge of the paper, just like that. And this is a really important skill for when you're using real microscope slide. Find the edge of the paper. There it is right there. Now I just take my coarse adjustment knob and I adjust it. Keep adjusting. There it is. Cool. I'm going to fine adjust until I get a good picture of the paper. Wow, that paper is kind of cool. See that? You can see the fiber in the paper. Oops, a little blurred. Let's slide it down just a little bit more. There you go. See the fibers? My autofocus is wanting to focus. All right. I'm going to find the hole. There it is. There's the edge of the hole. That's pretty cool. Look at those fibers. Huh. It's like a little city there. See the fibers on the paper? I can follow that hole around. Up. Over. Oh, there's part of my picture. Right now I'm right above. So I'm going to start over here. There's the circle. I'm looking for the orange right above. You see it? There's cut out right above. You can leave your paper in. I can find this easily again. Because I pulled out at a certain magnification, I'm putting it right back in at the same. So there's the top of the circle, and there's the tiger. Nice and orange. Or not. What happened? Why don't I see any orange? I don't see any orange at all here. I see yellows, and blues, and reds, and blacks. Looks like occasionally green where I have a blue and a yellow overlap. What is going on? I'm way into the tiger now. Still nothing? It's crazy. No orange. Oh, maybe I see a little bit of an orange where a yellow and a red overlap. There's some black. That must be a stripe. Totally a stripe. Look at that. Yeah, that's a stripe on it. Still no orange. I just scanned through most of the tiger's arm and not a drop of orange. Insane. Why is that? Well, here's why. Pointillism. So pointillism is actually a form of art where the artist figured out that if you take little dots of certain colors and you put them in close enough vicinity, as the person gets back far away, the eye will blend those colors together and make a color for you. So instead of having all the possible colors of the rainbow in a printer, you use 3 colors, 4 technically. Black, usually yellow, reds, and blues. So when they spray, the spray pattern of those little dots, or the print pattern of those dots, are spread apart just enough so that you can see a color. But when you get really close to it, you can distinguish them. It's called resolution. When I'm magnified up and close, I can resolve the difference between those colors, but my eyes can't tell when I'm further back away. So it's actually a really cool QR you can do on here. It's a Marilyn Monroe and Albert Einstein image, and I remember seeing this when I was in college. But when you look at it up close, you see Marilyn Monroe. You pull it back, and you see Albert Einstein. It's because the pattern of the dots at different differences or different distances make your eyes see different images. So it's really cool. But check it out. It's called pointillism. How about that purple? Purple is a pretty bold color. Let's try that. Edge of the paper, the whole, man, I love looking at the edge of papers. Look at that fabric. So cool. You can actually Google how to make paper out of lint at home. So you can take dryer lint and you can turn it into paper by flattening it out, and it looks a lot like that, and you can check it out under the microscope too. So look at the purple. The purple is so bold that those dots are so close together that right now I'm actually seeing purple. In fact, if I blur, it just looks like purple. But purple is not one of the colors. What I'm actually seeing are the reds and the blues really close together. So what I can do is take this, I fine-tuned it, right? As fine-tuned as I'm gonna get, and I'll flip it. Boom. Still looking pretty purple. And now I can really see how the reds and the blues, I have a bunch of red dots and blue dots, but when they're close together where they kind of overlap, I'm seeing a purpley color. But that's it. With a microscope, you can see things you've never seen before. You can look at how things are made. I mean, I would never really imagine that the printer didn't have purple and all the other colors, but when you refill a printer, you only see those 4 cartridges, the black, the yellow, the red, and the blue. And that's what it does, is it mixes all of those 3 colors to get the right combination for your eyes at a distance. Put that back, we'll go on to the next section. Love it. Let's look at some newspaper. So the next section, navigating the specimen. Have you ever noticed that things just don't seem to move where you want them to do when you're trying to slide things already? So what we're going to do is we're going to take a piece of newspaper. So you have a piece of newspaper in your envelope, and you're going to look for a letter E. Right? Lots of things on here. Excuse me. All right, I'm gonna look for a letter E that's closest to the edge. Let's do, thank goodness she joined because she possesses organizing. There are lots of E's in there. So I'm gonna take it. I'm gonna go back to my scanning, make sure it's the red ringed. I'm gonna bring this in. Kind of cheating because I know my focal point where I want to focus is actually right there where paper would be. So look at that right away. I already know. And boom, found an E. So I can see when I'm reading it, the E is upright because here's the print. I can read this whole article right here. Mutual aid organizations or grassroots groups, blah, blah, blah. I can read it. When I put this in, let me find that E. There it is. So I'm going to slide my paper to the right. I'm going to slide it down a little bit. There we have the E, right? So how do I go from here? Yeah, get used to it. A lot of questions because we're about to move into the next section where I'm going to ask you lots of questions to help you learn your scope. I'm going to turn the fine tune knob. Boom, there it is. Beautiful. So on my notes, what I'll do is I'll write down my observation. My question was, when I'm moving around on the slide, why do things not move the way they are? That's what I observe. My question is, if I look at letters, will the letters be organized underneath the microscope? So will it look like a normal letter? Is that what the normal letter E looks like? No, right? So my question is, will they look like the normal letters and will they move in the direction I expect them to? So if I move to the left, will it move to the left? If I move to the right, will it move to the right? So my hypothesis is, number one, is it's going to flip things because I've noticed that things seem backwards when I look in the microscope. So I think it's going to mirror and it's going to move in the opposite direction that I predict. That's my hypothesis. I'm experimenting. My experiment was I'm going to look at the letter E under the microscope, which I did, and I'm going to look at what happens when I slide it. So the first thing I'm going to do is my letter E on my page was a normal letter E. So I'm just drawing a picture of a normal letter E. But this is actually what I'm seeing. I'm seeing it mirror imaged. It's flipped backwards because the normal E's opening is on the right. The E I'm looking at has it on the left. So I drew a quick picture underneath here. I'm showing kind of the fuzzies. You don't have to be a super artist. You just want to note the details. It's kind of fuzzy around. I can see the material or the grains of the paper. My ocular lens is 10x. My objective is 4x. So my total magnification is 40x. I'm going to try this at the 10-10. 10 from the objective, sorry, ocular, and 10 from the objective. So I'm just going to rotate this. Your microscope is actually a pretty good microscope because it's par-centered, which means that the center here, center of the E, should stay pretty close to the center on the next objective. Where'd it go? Well, here's the problem. Your microscope's not parfocal. So parfocal means that it would stay in focus when I flip from one objective to another. It's not far off, so I'll just tune the fine, only the fine tune, and there it is. There's that middle bar of my E right here. So I was looking right here at the E as my center, and that's what it is. So I kind of notice the pattern. I can notice there's some fibers going back and forth. But I can see the dark, rough edges, like the print was sprayed on or something and it bled. I don't see the little splotchy dots that I saw with my color picture. So let's try it one more. Let's go ahead and flip this to the blue. Nothing. Oh, there it is. A little hard to see. And my automatic adjust is going crazy on my phone. But I can see the very tip of somewhere along here. Right, so I just blow that up and show that one up edge at 40, 10, and 400. 40 time objective, 10 ocular eyepiece, 400 times magnified. So I make my notes. Here are my notes. These are the things I observed. It's a mirror image, first of all. The edges are blurred. I can actually see the edge. It looks like fine detailed print on the paper, but when I look closely, I can see that it's just kind of sprayed and it leaks, right? When I move things to the left, they move to the right. When I move things up, they do move up. When I move things to the right, they move left. When I move things down, they do move down. So it's all about mirrors, it seems like, right? It's a mirror image left to right, but not up to down. I can see the fibers. Another note that's important is that where would you want to, if you were studying the letter E, where would be the best place to look? Would it be the strongest magnification? No. Sometimes you don't need a lot of power. Sometimes just using the lowest magnification is good enough. You don't need it all. Right? So you recorded those things. We noticed what we observed. You can also notice things like the paper. Let one question lead you into another question. Now I'm really curious about that paper and those fabrics and the direction, the fibers I saw on the hole around the edges, the fibers I saw on the newspaper, how they crisscross, the way that the print worked on it. So you can do text analysis. You can reset your microscope, study the paper. What I'm gonna do, I'm just gonna rip the edge off of the paper and I'm gonna go back and look. In fact, I'm gonna move my stage clips, which are these little springy things, see? Slide this in. Look at that, so cool. I'm just going to explore the edge of the paper. By doing this, just exploring, it's going to help you get used to the microscope. Look at those fibers, it's so cool. All right, I have it kind of where, I had it kind of where I wanted it. Yeah, there's some nice fibers. You put the stage clip down and it moves it again. So I can take a really good peek at those fibers. Very cool. All right, don't stop there. So I draw a little picture. This is a newspaper. I'm looking at 40 times magnification because there's a 4 objective and a 10 ocular. And then I can see there's a lot of pattern up here, crisscrossing. That'd be perfect. But I can really see here I have these stringy fibers. They're not exactly like string. They're almost, I don't even know how to describe them. Almost like frost, right? So I have all these frosty fibers. I have the top light on. I can always flip it to the bottom and see what they look like. Whoa, a lot different. Back here. Yeah, it's just too much. So remember the knob on the side? This turns the light down, the rheostat. Not making much of a difference. Yeah, so it looks like the only light that's really useful is the top light. Yeah, I dim that a little bit. It's nice because you can see a little more contrast, which is the difference between light and dark, which is kind of cool. I can take the edge of some other paper, take a bookmark, see what that looks like under the edge. Looks a lot the same. Almost looks like a sheet of plywood that you find at a hardware store. Huh. So those fibers don't seem as stringy as the other ones. So I would just draw a picture. Oops, went out of focus again. There we go. And there's one stringy part. Not perfect, but these are all shady in here. They're not as stringy as the last one. And then I saw the crisscross fibers again. And that's cardstock. Bookmark. At 40x. And what did I notice about it? More dense. Less stringy. Something I noticed. And I'll jump again. You know what? You have a paper towel. Why not try a paper towel? Whoa. Whoa, those are really spread out. Very spread out. Even deeper into it, they're still very spread out. Why would that be? So if I draw a picture of the paper towel, I had a lot of fibers, and I can see a lot of space. So I'm drawing the darker ones being closer to me, and then some lighter ones showing the depth. But that's how they were. They were just random like that. Why do you think that is? Well, here's why. If I take my marker, where did I put it? Right down here. If you take your marker, experiment with it. Put it on the newspaper. So I'm going to take the marker, my newspaper in. I'm just going to clip it into an empty area, focus it. Just as an example, I'll put my tip in. There's my marker tip. Going down. Look at it spread. It's so cool. Look at the ink spread. It's moving from one fiber to another fiber to another fiber. Bled through. Let's try the same thing with the paper towel. So we'll clip the paper towel down, focus on it, bring my marker in. There's the tip. Oops, lost it. There it is. Push it straight down here. Oh, look how fast that moves. A little bit faster. Way faster. And I can still see the wetness of the ink because it's actually soaking the ink up. It's pulling the ink. All that space between the fibers is sucking the ink in, and it's actually filling the space in between with wet marker. So it can pull it faster. That's what makes this the quicker picker upper than the piece of paper, newspaper. But don't stop there. Keep exploring. Keep trying different things. See what happens when I do the same marker experiment on a piece of tape, or if I do the same marker experiment on a piece of magazine paper. Look at the different textures. Be curious. Try it. You can analyze different papers and see if you can figure out what makes them so special or different. Look at some old papers. See if you can find an old piece of paper from your family from 50, 60 years ago or whatever. Don't rip it, don't cut it, but slide the edge under the microscope and see if you can tell the difference between aged paper and new paper. In fact, I put a little blurb there on the bottom that talks about the Declaration of Independence. Someone found an original copy, which seems like an oxymoron, an original copy of the Declaration of Independence in the back of a picture frame that they bought at a garage sale for a couple bucks. And that copy had to go through all the analysis to make sure the date was right, for when the paper was made, the ink was the right kind of ink, the signatures were the matching, and it was crazy. But just try signing your name on a piece of paper and then make a photocopy of your name. Look at the difference. Can you tell the difference under a microscope, which one's which is the original, which is not? But do that, experiment, get crazy, and take a break, and I will see you on Microscopy 102, where we'll actually learn how to do slides, how to build slides. We'll look at bugs under the slide, we'll look at some rocks and a lot of really cool stuff under the slides, but it's more exploring with slides where you just been learning about basic parts of the microscope so far. We'll talk to you soon.